am here with Paul Hudson, Associate Professor at the KTH, the Royal Technological School here in Stockholm.
Yeah.
So you are a chemical engineer by training.
Yeah, that's right.
And doing synthetic biology.
Yeah. It seems like they don't match up, but they do kind of, because there's this thinking now that the cell can be thought of as like a little factory.
Yeah.
And with pipes that are connected. These are the reactions, and they're connecting metabolites and things like this. And so there's a mathematical description of the cell, which chemical engineers are quite comfortable with.
Because synthetic biology, what is that actually?
Yeah, well, it's quite a nebulous term actually. So I think synthetic biology, I would say, is mostly about controlling the cell metabolism. It takes native transcription factors or things like this that are in the cell that are responsive to certain stimuli, and it hooks them up in new ways. So we can make GFP turn on when you add a sugar or something like this to the cell. So it's really about controlling cell metabolism, I would say. And the synthetic part is that it often requires a lot of genetic engineering to do that. So there's other concepts like synthetic metabolism, and that's a bit different, but synthetic biology is mostly about controlling.
So it's making, it's like making cells do what we want.
Yeah, exactly.
Not necessarily what they want.
No, right. And this turns out to be a lot harder than it is on paper, actually, because the cell wants what it wants, and it's evolved to do what it wants. And we come in with our ideas, which are often not compatible with what the cell wants, of course.
Which, of course, brings us to, you study a cyanobacteria, which we may known as the blue-green algae. We see it every year almost in our waterways.
That's right,
and lakes. And what are you trying to make them do?
Yeah. Well, all right. So the cyanobacteria, the blue-green algae, there are a lot of interest in biotechnology right now because they do photosynthesis, and when they do photosynthesis, they capture carbon dioxide from the atmosphere, and they convert it into, well, more cells. We're trying to get the cyanobacteria, instead of making more cells, we're trying to get them to fix carbon dioxide and produce something useful, like chemicals or fuels or something like this.
Yes.
Yeah. Which they don't like to do. This is not what they're meant to do.
It's not what they're meant to
No.
do.
Right. So with synthetic biology, what we can do is with cyanobacteria, is that they don't produce these chemicals naturally. A cyanobacteria produces only more cells. That's all it makes. It secretes almost nothing. Whereas some bacteria, or let's say some fungi like yeast, they secrete ethanol in some situations, right? So we think one way to think of it would be, how can we create a cyanobacteria that secretes ethanol like a yeast does? Yeah. And so the first thing we have to do is we have to take the genes that are in that yeast and put them into the cyanobacteria, because it's only a few genes to synthesize ethanol, but cyanobacteria don't have them.
No.
Right. So if we do that, the cyanobacteria, it's amazing because they can read DNA from almost anywhere.
Okay.
Yeah.
So it doesn't have to be their own.
No, exactly. And this is like a key tenet of synthetic biology, is that they say what works for the elephant works for E. coli. So it's like, it's just DNA, So
they can read any DNA that you put into it.
Right. So I mean, this was discovered back in the 70s. I mean, this won the Nobel Prize, this recombinant DNA technology. Yes. And it works also for cyanobacteria. So you can take DNA from other organisms, you can put it into cyanobacteria, and they tend to read it and they express these genes. Yeah. So go back to our yeast example. They can make a little bit of ethanol, but they don't make a lot, right? They make it's like nothing to write home about, right? But it's still like a proof of concept. Is quite amazing because they're taking carbon dioxide and they're converting it into something that's much more energy dense. I mean, carbon dioxide has no energy in it really, but ethanol has some energy. So it's like, when we see that biology can do this process, I mean, this would be a very good chemical process. If chemical engineers could figure this out to do it synthetically. I mean, I guess they do with this Fischer-Tropsch and everything, but it requires lots of energy. Now, cyanobacteria do it with just the sunlight. Yeah. With carbon dioxide from the air. I mean, it's amazing. They take air. It's just like if it was sitting right here, it could make ethanol. It could make a little bit. It's very small, but it's like fuel from nothing, it feels like, right? Yeah. So that's really the promise, right? The problem is that it makes so little that it's almost impossible to even separate it, purify it. It's not worth economically. So then where synthetic biology then needs to go is to how to get them to make more. Yeah. So that, like, just getting the demonstration, that's synthetic biology 1.0. and now we need to go to like 3.0. We have to take many more iterations of complexity to get this to work.
Yeah. Because you have worked a little on how much that it needs to be improved almost to make it feasible.
Yeah. 100-fold, I would say. I think we're at like 1% of where we should be. Yeah. And you know, the theoretical limit. So you see, there is like limits to what photosynthesis can do. And this is one critique about cyanobacteria is that, for example, they're green. So that means the green light is not absorbed. So they're not perfect, right? They're not perfect converters. But the theoretical limit, it should be economically viable. If we can get them to produce near the theoretical limit. Which we're far from, but I think we're maybe, I mean, like I said, 1% is a little rough. I mean, probably 5%. We're probably 5% of the way there. But I think there's a path forward. I mean, I believe that, and that's why I'm working on this. One inspiration, is definitely finding new energy solutions. I mean, I think it's very important. I don't know if cyanobacteria will be the final solution. I mean, maybe you can edit that out too, because that has bad connotation.
That's not really what we want here. Will be the best solution.
Yeah, exactly. The best solution. I don't know if it will be. It'll probably be. It can be like a mixture. It can contribute, right? So one thing about biofuels is that it's so hard to compete against oil. I mean, oil now, as you're taping, is $20 a barrel or something. It has not been this cheap in 20 years.
And the energy density is...
Yeah, the energy density of oil is 50% higher than ethanol. And it's just like, it's insane how cheap they can produce oil. Usually when you produce things with biology these days, you think that you produce complex things with biology because biological chemistry is very complex. So we're trying to produce some of the cheapest thing known. And so it's very difficult to compete on price. But it has to be, because I think competing on price is the only way for it to get widespread. Oh, he's fine. Yeah, so that's one motivation, is to really find some sort of... I mean, when I was growing up, there was all this talk about why we were in Iraq. Like, I remember this, and it was like, oh, we want the oil. And it's just so terrible to think about all the war and stuff that's caused by oil. And if we can use sunlight, anyone can use that. Sweden can use it. The Middle East can use it. It doesn't matter. Like, I think that's really that's a strong motivation to just, when I was growing up, is to get in these freaking wars. I'm sorry it's so political, but it was really like a driving thing for me. Like, energy independence, I think, is very important for country, for people. So that, I would say, energy independence is a very important thing, and I believe that. So.
And that's sort of still what makes you, even though that you feel that there's maybe a 5% and 100% may not be around the corner, but there is that sense of it needs to happen.
Yeah, I think so, and I think, back when oil was expensive, maybe 10 years ago, there was a huge push to get to, in science about cyanobacteria, and that got us essentially this far from nothing. So I think that it is. Now, when oil prices drop, then funding goes away magically, right, for biofuels. So sadly, there was a lull. Now, but that's starting to pick back up again. We got some new breakthroughs that are making it more easier to do genetic engineering in these things. So I think that it can work.
Where's the limit in synthetic biology? I mean, now we talk fuel, which is a very hands-on approach.
Right, yeah.
But if we can get them, like bacteria or yeasts or any microorganism, to produce whatever we want, is that feasible? Is it possible?
Yeah, I mean, it almost feels like it's possible, actually, to do it. I mean, every week there are new papers coming out. I mean, I just saw that they produce morphine now in yeast. I mean, and that's a really huge advance, because it really showed that you could take these complex plant molecules, and morphine is incredibly complex, synthesized chemically. Probably impossible, I don't know for sure, but I'm thinking it's very difficult to synthesize chemically, and now they have a yeast that can produce it. I mean, again, some of the problems with the biology is that it can produce it, but it doesn't always produce that much. So I mean, it's always like getting it to be economically viable, which is very hard. But I mean, the demonstration, we can produce almost anything with biology, is true. I mean, amazing reactions that are discovered every year, new reactions that enzymes can do. So I think that's the future. So you were asking about, communicating about synthetic biology to society. And I remember that we had a paper, we had just had cyanobacteria that produced butanol. So small amounts of butanol, but it was a good, I thought, proof of principle. And there was a little article written in Aftonbladet, which is like the daily, a nightly newspaper in Sweden. And I saw that article, and then they had the comment section open and I just thought I would look at what the people were saying about this, and there were so many comments about if the cyanobacteria were to escape. Like these were genetic engineered cyanobacteria, so they were GMO, and had the scientists, they never think about the implications if the cyanobacteria were to get out, right? Like what if it gets out into the ocean and then pollutes the ocean somehow? And so I thought, well, this is maybe a failure on my part to talk about this at all, to the press. I mean, I didn't mention it at all in the interview or anything like that. So I think, I mean, by the way, there is really very small risk of this happening because these cyanobacteria that produce, when they produce biofuels like this, they barely grow. They're so unfit that they probably could not survive out in the wild anyway.
They are not competitive.
No, they would be out-competed somehow. But I understand, I mean, of course, it's not allowed to put GMO right now. So they can't even be grown outside. It's not even allowed to grow them outside in Sweden. They have to be in a laboratory, which they are. So
we are able to manipulate biology.
Come here.
And what does that mean?
Well, yeah, I mean, I think one immediate implication is that we can cure disease that are simple, easy to understand diseases. So diseases that we know only have one nucleotide change. Yeah. That's really
that's the hope.
Yeah, that's amazing. Yeah. But the problem is that we start wielding things we don't understand. I mean, we see this even when we work with cyanobacteria. Now, bacteria are very simple. It seems like we know everything there is to know about bacteria. Well, it's not exactly true because, of course, they behave in unexpected ways when we start to manipulate the genome. And that's our research. We think we have a plan to manipulate the genome. We do that. We check if it went according to plan. Often it does, but sometimes it does not. And then we need to reevaluate our decision and try again. So we do a cycle. We design, we build, we test, and we learn. Right? This is the cycle.
Absolutely.
But that's very difficult to do outside of the bacteria world, right? In organisms that are not bacteria, it's impossible.
And I mean, it's also, we are wielding things that we don't understand. We're just scratching the surface of biology.
Yeah.
It is, like you say, it's the next frontier.
I mean, there was this thinking about the human genome, that the human genome was full of what they call junk DNA. So I mean, that only 2 or 3% of our genome did anything useful. And now that's starting to be questioned because now we can interrogate different parts of the genome. I mean, you worked in medicine, right? So you understand this also. And it turns out that a lot of this junk region, they actually did stuff. And so, previous gene editing back in the 90s, it turned out that gene editing was changing some of these regions of junk DNA. And then people got really sick. So there was a moratorium on gene editing for 20 years or something. So... and now we have even better tools. So it's like now it's even easier, but our understanding has increased, yes, but probably not at the same rate as our ability. Right?
Exactly. So technology has sort of increased faster than maybe our understanding.
I think so. Yeah.
I think there's something about the technology right now that is going incredible fast.
Yeah. Even in my field. I mean, I now it's grant writing season, and I think, looking like how much has changed. I'm reviewing papers and stuff. How much is changing and so fast, with synthetic biology. I mean, 5 years ago you could barely do synthetic biology in cyanobacteria, and now it's like an explosion of different promoters and things that you can use. So I'm thinking, and of course, then I start to get new ideas because now the technology allows me to change things that I couldn't change before. So it's so fast. I mean, really like an exponential increase in just this field. It's amazing. What is the magic bullet to get these cyanobacteria to produce more of what we want? So one particular challenge is that if the cell is making a lot of ethanol, let's say, or product that we want, they tend to mutate this burden. It's a burden for the cell, and they tend to mutate the genes away. So there's native defense systems that mutate the genes. And then the cells, they revert back to non-producer phenotype. And so I think this is a really big problem for us because using synthetic biology, we can divert almost the entire metabolic pathways to producing ethanol. So it's almost like 100% conversion of carbon dioxide, but it doesn't last. because the cells, if you have one cell in that population in the reactor that mutates, it will grow faster and take over the culture. So I think this is the number one challenge, is that how do we get them to do this for a long period of time to produce these compounds?
So thank you for coming out for a walk and talk.
Yeah, thank you for having me. It was very nice, actually.
It was very nice.
It's a good thing it wasn't yesterday.
It was. It's raining.
Yeah.
All right.
Bye-bye.
Bye.